Hello and a warm welcome to everyone to us today's webinar on biomarkers and endpoint assessments in COPD complex generic style. My name is Bhavika Maheshwari and I represent Veeda Group. Veeda Group is a full-service contract research organization offering comprehensive end-to-end solutions ranging from preclinical to clinical and bioanalytical services to support innovator, biosimilar, and generic drug development programs for our global clientele. We have significantly expanded our global presence with an extensive clinical site situated across 26 geographies. Together, we serve clients globally in pharmaceutical and biopharmaceutical industries, agrochemical and industrial chemical industries, herbal and nutraceutical industries, medical device industries. As your facilitator today, I'd like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by a Q&A round. We have enabled our Ask a Question feature. It's right on your screen where you can drop all your questions related to this webinar right in there. Now, moving forward, I would like to introduce you all to our panelists for today's session. Dr. Hiren Mehta. Dr. Hiren Mehta is a PhD in clinical pharmacology with over 19 years of experience in managing clinical pharmacology functions, medical and scientific affairs. Dr. Mehta has worked with renowned companies such as Alta Sciences, Sinus Health, Lambda Therapeutic Research, Torrent Pharma, Cadila Pharmaceutics Limited, and Bayat Ledol Limited, where he has demonstrated expertise in pharmacokinetics, pharmacodynamics, data analysis reporting, and regulatory compliance. Our second panelist for today's webinar is Mr. Paresh Patel. He has approximately 13 years of rich experience in the CRO and pharma industry. Mr. Patel specializes in clinical research for various studies involving different dosage forms, including oral dosage form, lyophilized dosage form, transdermal patch, topical dosage form, nasal or oral inhalation dosage form, long-acting depot injection, and solution for IV bolus or infusion injection. His expertise extends to both synthetic and rDNA recombinant drug products in both healthy and patient populations. So, without any further delay, I'd like to virtually call upon Dr. Hiren Mehta to begin this session.
Thank you, Bhavika, for the nice introduction. Good afternoon, good morning, everyone. At Veeda, I look after the various scientific affair operations, including PK-based data management and medical writing. I will quickly jump to the presentation and share my screen. I hope it's visible on the screen at the moment.
Yes, Dr. Ray.
Okay. Just give me 1 second. Okay, so I will start with the epidemiology of the respiratory disease. The respiratory disease constitute a huge burden in our society. Worldwide, there are about 235 million people living with asthma, around 251 million with chronic obstructive pulmonary disease, which is COPD, and around more than 70,000 people with cystic fibrosis or CF. Around 3 million people are affected by idiopathic pulmonary fibrosis, IPF, and around 10 million with contract tuberculosis every year. The global respiratory drug market currently grows at an annual rate between 4 to 6%, which is quite high. And most of these indications, they mostly try to treat with the orally inhaled drug products or OIDP. We'll use this term OIDP frequently during the session, which usually means all the inhaled drug products used for respiratory disease. Now, there are certain advantages of the orally inhaled drug products. Few of them will be the delivery of the drugs directly at the site of action. Because of directly at the site of action, they also offer faster onset of action. And a very small dose can be effective compared to the oral or any other route of administration. It is painless, of course, and improving patient compliance, therefore. Also, it has a lower systemic concentration and hence lesser the adverse drug reaction. It also offers an absence of first pass metabolism because it is bypassing the gastrointestinal tract. Certain advantages of OIDP. Now, I'll give you some examples of OIDP that normally used for the asthma and COPD. That includes beta adrenergic agonists like albuterol, formoterol, etc., and few muscarinic antagonists, ipratropium, tiotropium, including, and they induce bronchodilation. Other glucocorticosteroids used to control the inflammation includes fluticasone and budesonide. For cystic fibrosis, they normally use mucolytics, which are saline and acetylcholine. They normally use to thin the mucus of the trach, facilitate the clearance from the patient's lung. And alternatively, they sometimes also use leukocyte DNAs to reduce the inflammation and some antimicrobial agents in severe conditions as tobramycin. In the next slide, I'm planning to demonstrate how the orally inhaled drug products disposition occurs within the body once someone takes the drug administration using the mouth and the pharynx. In the upper part, you will see that a person taking the dose using the canister, out of that total administered dose, around 10 to 60% they deposits in the lung using a fixed factor. And from the lungs, it is completely absorbed and goes to the systemic circulation. Here, there is no wastage of the drug and it goes directly into the systemic circulation. However, from the administered dose, around 40 to 90% are being swallowed. Maybe it is reduced by using a spacer or mouth rinsing, but in general, for more than 50-60% dose is being swallowed directly into the GI tract. From the GI tract, it goes to the liver for first pass, and then it goes to systemic circulation. Some drugs who are highly bioavailable, they have some systemic side effects even after the respiratory route because of the direct disposition and absorption from the lung. These are some of the examples of the orally inhaled drug products. The first two on the top line are normally very much patient friendly, and most of the patients prefer to use it. Starting with the meter dose inhaler or MDI, it's an aerosolization solution, a non-aqueous formulation, but it has a canister. It offers fast aerosol delivery, and the API can be suspended or in a solution form, but in general, it is non-aqueous. It typically deposits as dry particles, and sometimes it is little bit wet, but normally it is dry particle directly on the site of the axon. The top right is dry powder inhaler. It is again very much patient friendly, and um, it also offers aerosolization. It can be in blister, capsule, or reservoir presentations that can be directly used inside the device and blend it. It's a mixture of API and a carrier like lactose, either in the form of particles or agglomerates. It also deposits a dry particle of drug or agglomerates at the site of action. The last two on the bottom line are normally used in hospital setup. Sometimes patients directly use it. The left in the corner of bottom normally used in the hospital setup where we normally call it a nebulizer. It again offers the aerosolization, and the product is normally aqueous with some with using of the ampules. The API, however, can be suspended or in a solution form. It also deposits as droplets, and it contains the dissolved or the suspended drug. The inhalation spray is again a patient-friendly device. It is an aqueous formulation coming with a cartridge. The API is in a solution form, and also it deposits as a droplet, as a bar, and the drug is dissolved inside the solution. Now, most of the generic companies who are focusing on developing the orally inhaled drug products, the indication most likely will be the respiratory disease. And it's not an easy task to develop an OIDP compared to other solid or oral dosage forms, because the OIDP development currently is suffering as an overall attrition rate of 70%. The cumulative probability of an orally inhaled drug products to reach the clinical market is only up to 3% compared to 6 to 14% of other diseases. So you can see how difficult is it to develop the orally inhaled drug products because of multifactorial problems and several contributing causes. Normally, the problems the companies are facing are in general, poor understanding of the underlying mechanism of respiratory disease itself. The formulation itself is a problem and challenge in terms of how to decide the particle size, the distribution rate, the solution, the suspension or dry powder itself, and also the poor performances of the drug administration methods by patients or technician or sometimes clinicians also. All of these make it very difficult to develop an orally inhaled drug product. Now, the mechanism of the oral inhaled drug products are at multifactorial, and the disposition happens at the target site after first administration. So we'll talk in a sequence of delivery, disposition, and then elimination. So the delivery or the disposition of the product itself is dependent on two main parameters of the particle size of the particles or droplets which is carrying the drug. First is the velocity, and the second one is the aerodynamic size distribution. This is a physicochemical property of the product which contributes a lot in delivering the substance at this site. Now, after the administration at the site, how the disposition occurs, it also influenced by presence of lot of things, including mast cells, dendritic cells, Clara cells, and macrophages. And the protease activity also can reduce the bioavailability. So this is about the disposition in lungs after we administer the drug. After that, it is getting eliminated from the systemic circulation, also from the site of action. And the contributing factors are the mucociliary clearance pathways, the macrophage clearance, and the drug clearance by an absorption itself. So if you see, there are three-tier complexity in overall mechanism of the product, especially for orally inhaled drug products. The delivery itself is a complex phenomena, followed by a disposition in lungs and elimination of the product. Now, we all agree that it's evident now that then OIDP development is not an easy task. The success of OIDT development depends on mostly the understanding of the features involved in the disposition of the product itself at several levels that we discussed in the last slide. Sometimes a predictive preclinical model can also bring a success when we are developing a new product. In last 4 years on clinicaltrial.gov, there are more than 2,500 inhalational clinical trials against total of more than 97,000 total trials. So which basically means only 2.6% of total trials are inhalational clinical trials. This also narrates how difficult it is to develop the orally inhaled drug product. And more than 1,000 different inhalational conditions and endpoints have been logged among all these clinical trials. It is also important to observe that more than half of these inhalation studies are for systemic conditions, which are beyond the domain of respiratory disease. So the point here is, though they are developing a product of that nature, orally inhaled drug product, however, not all the time they are for respiratory disease. They may be for systemic conditions sometimes. So let us talk about once the formulation team decides to develop a product and come up with a prototype and talks to the clinical team about what are the required PK or in vitro or in vivo or pharmacodynamic studies for such a product approval at different regulatory agencies. I will briefly discuss in my slide and then hand it over to my PK expert Paresh. So normally the common approach for this kind of product is pharmacokinetic studies to determine the bioequivalence, and it is suitable for all systemically acting products. Now, this may not be sufficiently possible to establish a bioequivalence of locally acting drug products only by the pharmacokinetic endpoints. So the alternate studies for locally acting drug products, especially the inhalation products, can be in vitro. There are several options in vitro studies that we will discuss later. And in vivo PK study that we think it's is must to check the systemic levels, and also the pharmacodynamic or a clinical endpoint study. So these are the three different type of variety of studies that we may have to conduct to seek the regulatory approval. I will pause here and request Mr. Paresh to take on further slides for regulatory specific requirement for different drug products. Over to you, Paresh.
Thank you, Dr. Hello. Good morning to all, and thank you, Dr. Hiren, for allow me to present subsequent slides. And in today's discussion, generally we focus for the two regulatory main. One is FDA and another is European regulatory requirement. So firstly, we cover FDA requirements for the generic applicants to require for orally inhaled drug products. So generally for generic applicant, it's called ANDA submission, and for the generic product approval, we have to this is orally inhaled drug product which are combination of drug as well as device itself. So first we have to show similarity between device. So either it should be similar or deemed to be similar, because certain times it's have a restriction based on the patent. And second, we have to prove in vitro performance similarity across test and reference product. Third one is we have to save similar safety by considering PK endpoint study, where we have to show similar systemic exposure. And lastly, we have to prove similar efficacy via therapeutic equivalence or clinical endpoint study between test and reference as a superiority against the placebo. So generally, this is the decision tree for the FDA, where we have to follow all the component to prove similarity across device, in vitro test, PK endpoint study, as well as clinical endpoint. If we are meet all the four criteria, then and only we can able to got the product approval as a bioequivalence with the reference product. So it is called the weight-based approach. So this is the generally requirement for the in vitro test, where the first we have to prove Q1, Q2 sameness between test and reference. Second, we have to perform certain in vitro B studies like single actuation content, drug in small particle droplet, plumb geometry, drug particle size distribution, droplet size distribution by laser diffraction, spray pattern, priming, repriming, and dissolution. Once we prove biosimilarity in this in vitro assessment, then we can have to prove bioequivalence in the PK endpoint studies. And lastly, we have to prove the clinical endpoint bioequivalence study. So this is the basic study design required for the PK endpoint bioequivalence study. And the most common design generally used is two-way crossover design. And we have to check the systemic exposure between test and reference product. And for the strength to study, generally for the orally inhaled drug products, regulatory as of now lack of information about dose linearity and proportionality, and lack of information about in vitro performance and correlation with the drug behavior. Regulatory insists to perform the PK endpoint study for all the strengths, and those generally minimum number of inhalation required to characterize the PK, and using sensitive analytical method they recommended. So the selection of number of inhalation should be based on stepwise manner. Like in first you can attempt one or two. If you are not get the sufficient PK profile, you can increase to the three or four. But it should be the within the safety limits of the labeled daily dosing. And generally, FDA recommended this study in the healthy male and non-pregnant female population. And the bioequivalence endpoint for the PK endpoint study, generally 90% CI should be fall within 80 to 125% for standard Cmax and AUC parameter as a PK endpoint study. Now we are talking about the clinical endpoint, PD endpoint studies requirement for the generic inhaler product. So generally the main study design would be the placebo controlled randomized parallel or crossover clinical endpoint study or pharmacodynamic BE study. And generally, this study involve placebo run-in period where there will be no any treatment given to the subjects, and the previous continuation treatment should be washed out during those run-in period. And to check the placebo effect, studies subjects should be selected based on the non-responder to the placebo arm. And objective of this study is generally to determine the local exposure and local effect of the drug at the site level between test and reference. And in clinical endpoint study also, agency emphasis to perform at lowest dose only. And the dosing should be either single or multiple dose based on the mechanism of action of the PK or disease state. And this study we require to perform in the patient population where the product is already approved. And the endpoint for the clinical endpoint study, generally we have to prove the bioequivalence between test and reference considering 90% CI ratio of clinical or uh, PD endpoint biomarker within 80 to 125%. Or for PD endpoint, we have to prove relative viability. 90% CI should be fall within 67 to 150% using the dose response model. So for orally inhaled drug products, lots of challenges and complexity are involved as compared to the normal oral BAB studies. So for the dose selection is one of the complexity and challenge involved in the oral inhaled drug product, because systemic concentration is very low, and sometimes it's also affect to measure the drug level via bioanalytical method currently we have. Second is subject selection. And for the PKB, generally, we require healthy subjects, non-smoker subjects, while for the patient and clinical endpoint study, we require to enroll target disease patient, which is also challenging factor. And su- subject and patient required a training. So for the proper conduction of this training, we require to train subjects as well as patient prior to enroll in the study, and we also have to keep a record diary for the daily base of dosing at home and consider for the study duration entire. And there is a certain factor which are related to device, because the patent limitation may leads to the difficulty in the understanding of design between test and reference, and it also leads to some complexity to the patient for the daily dosing. Now we will discuss about the one of the case study which is belongs to the clinical endpoint study for the one of the molecule. So in that study, the study design was randomized multiple dose placebo control parallel group design, and treatment duration is 2 week run-in period with the placebo, and once placebo run-in period is over, 4 week treatment period is started in which test reference and placebo will be administered. And for the clinical endpoint study, generally huge sample size are required, and in that study around 800 to 900 patients are enrolled, and in the proportion of 10 gem 10 gem 1, where test products reference product administered as a 10% proportion while placebo only 1. Primary endpoints are area under the serial, FEV1 time curve from time 0 to 12 hour post dose, which is called AUC 0 to 12 hour on day 1 of the treatment. While second co-primary endpoint is the trough FEV1 on last day of the treatment. And the bioequivalence criteria for this study was to estimate the 90% confidence interval for the ratio of T by R of FEV1 of the first one, AU 0 to 12, should be fall within the 80 to 125%, and secondary, the co-primary of FEV1 trough values should be also fall within the 80 to 125%. And we have to prove superiority with the placebo at the alpha level of 5% for both test and reference product. And this is the disposition summary of the trial, where there is a huge screen failure. So totally 1526 patients were screened in the entire clinical trials, from where 647 screen fail, almost 42% of total sample size. and 239 patients having a predicted FEV1 between 40 to 85%, which is the primary inclusion criteria, and greater than 40% are not reported, and 168 are not meeting the inclusion exclusion criteria. So totally in the study for test and reference arm are almost 400, 118 and 419 patients were enrolled for test and reference, and 42 patients were enrolled in the placebo arm. And among them, 23% further discontinued from the test treatment, and from reference, almost 13 subjects were discontinued from the study, and 5 for placebo arm. So total 395 patients completed test treatment arm, 406 patients completed reference treatment arm, and 37 patients completed the placebo arm. So now this is the endpoint of the study where we have to measure the baseline FEV1, and post baseline we have to collect the over 0 to 12 hour FEV1 reading on day 1 of the treatment start. So this is the graphical presentation of our results where baseline subtracted FEV1 for the placebo, test and reference treatment arm, as showing that for placebo the marker, round marker with the no filling, it's having a low FEV1 value. Then subsequently red and black color are showing for the test and reference, where they are almost having a superimposable profile for FEV1. And another side we have presented the data of the trough and day 1 FEV1 90% confidence interval limit for the study. So basically study conclusion is that 90% CI of FEV1 of day 1 fall within the 80 to 125, and also the trough FEV1 of last dose is also fall within the 80 to 125, which showing that product is bioequivalent. And the all p-value for the placebo with the test and placebo with RLD also less than 0.05, which prove the superiority of test and reference with the placebo treatment. So these all are the requirements for the FDA regulatory requirement. It's called a weight-based approach. Now we will talk about the European regulatory requirement. So for the European regulatory requirement, submission is called the hybrid submission, and we have to prove device similarity as like US market, and also we have to prove the similar in vitro performance versus the European reference product. And for the safety similarity, generally European agency want study under PK endpoint with and without charcoal blockade. So for the drugs who have a more systemic viability using GI tract also, in that scenario, agency requested to perform the charcoal block study in addition to normal study. And for the local side effect action, product we have to prove equivalence with charcoal block and without charcoal block. So generally this is the approach where Europe is following the product approval. And step 1, generally if we prove the in vitro similarity, formulation similarity and device similarity, then product got the approval from the European agency. If we have any dissimilarity in any in vitro test or device, then we have to go for the PK endpoint study. So it is called step 2A, where the lung disposition study we have to perform via PK endpoint study. If we meet the PK endpoint study, then we can got the product approval. But if we are not proved the bioequivalence in the PK endpoint study, then we have to go for the step 3, where PD efficacy equivalent we have to prove. So this called the stepwise approach, where either of the approach we can prove biosimilarity between test and reference product, and we got the product approval. So for the European market, generally PK endpoint study is similar to US, but over here main difference is that we have to prove bioequivalence with and without charcoal block. So for that, either we can perform 2 separate study for the with and without charcoal block, or we require to perform 4 period combined study, where first 2 period we can have it with charcoal block and another 2 period we can have a without charcoal block. And the objective of the study is mainly to determine the difference in overall systemic or site level exposure of the drug products. And strength selection is based on the single lowest strength where the you can able to measure the drug level in the systemic circulation. And dose is depend on the inhalation strength, and minimum inhalation we require to dose for the healthy subjects. And population in the study is healthy male and non-pregnant female. And B endpoint criteria is for without charcoal block. AUC and Cmax parameters should be fall within 80 to 125 limit for geometric mean ratio, and it's 90% CI. And for with charcoal block endpoint, similar AUC and Cmax parameter, 90% CI T-by ratio should be fall within 80 to 125. And certain time, if we require to prove partial AUC bioequivalence to check whether the drug present its site of action in early minute, like in 30 minutes or 25 minutes, the observed systemic circulation is mainly because of the lungs deposition of the drug. So in that scenario, we have to show bioequivalence for the partial AUC like 0 to 30 minutes. And for PD equivalent study, generally Europe does not have any specific requirement, but they are following the similar study design endpoint assessment as per the European US FDA recommendation. Now, I will ask Dr. Hiren to continue for the subsequent slide.
Thank you, Paresh, for a nice introduction and an elaborative understanding of the FDA and EMA requirements to submit the generic product dossier. Now, that is all regulatory requirement. However, CROs and sponsors are sometimes find it very difficult to conduct these kind of studies because there are multiple challenges in conduct of these kind of studies due to several factors. I will walk you through in a brief way what kind of challenges we can face. There can be site-specific challenges about, lack of awareness about the spirometry in general for clinical trials. They may be using it for clinical practice, which is a very inaccurate or sometimes not uniform, but in trials we need it very uniform. A lack of calibrated or validated equipment for spirometry measurements. Quality of spirometry itself, as per the American Thoracic Society or European Respiratory Society requirements, and they have to go for a large patient enrollment, as we discussed in one of the case study for FDA, more than 42% skin fill ratio. So that is site level problems that sponsors and CRO are facing. There can be study specific challenges also because we always need to have a rescue medication, because for clinical endpoint or the pharmacodynamic study, we need patients. The site staff are not, not always available. So they maybe need to stay back for the long duration until the safety window is complete. Also, validity limited to self-administration only. We need to extend that to all the subjects, or including the technician should be able to extend the expertise to all the subjects. Sometimes the run-in phase using the placebo or the ongoing treatment is very long and extensive. And because we need to have a repeated serial spirometry measurements, sometimes home-based monitoring is also required, and that is causing a compliance challenge. In terms of the problems that patient may face after participating in this kind of trials is because there are various factors such as environmental, smoking history, genetic factors, which they do not allow the patient to participate in the trial, or they may have to withdraw themselves. The education and training of the patient is also required, at least for the spirometry measurement and how to use the device. The regulatory requirements, the patient may have to maintain symptom diaries, and they have to master the exacerbation capture technique using the device for accurate readings. The current medical practice on the top bottom corner is also a challenge for some sites because they have to keep the patient's medical records for future audit and source data purpose. How they used to medically manage the patient before the trial and now during the trial, and they have to also accurately describe all the adverse events and how they manage the adverse events. So these are the challenges in general for the site, for the sponsor, for the patient, for the clinicians. However, a proactive approach by the site, by the CRO or the sponsor can give us very good success ratios. I'm not talking about the study pass or fail, but at least get the accurate outcome of the study, which is supposed to happen after a 100% protocol compliance. One of the most important approach which can be proactively initiated is training, because the training of investigators and study site personnel is very important in this kind of studies, especially how to handle the device, how to record the assessments of spirometry, how to log them, how to train the patients and the reader. Also, the local logistic for sample and other trial material from the site to the central depot is also equally important. The site infrastructure itself also plays a very vital role, because of the equipment is always at the site, and different site will have different types of equipment unless until we ask them to use the same make and model. So we sometimes have to supply the equipment from our side to maintain the uniformity. The validation of the equipment is also required at all the sites for IQ, OQ, and PQ for the spirometer. And after the reading is done, the central upload of spirograms to a location where a reviewer can review them all together is also equally important. So the infrastructures along with the training at the site plays a major role. The shipment, however, is a common requirement for all kinds of studies. The calibration of digital clocks is equally important, because we have to measure the FEV1 and the volume at a specific time in a serial manner. So calibration of clocks is equally important, and the spirometer itself should be calibrated properly to give us uniform and reproducible results across all the sites for all the subjects. Now, this slide talks about how we can train a patient to master the inhalational technique, so that every time before the study, which is a baseline, as well as after the dose, which is post-dose, they give uniform and reproducible results, and we maintain a uniformity across all the sites and all the subjects. From the top left, we'll talk about the preparation, uh, how to prepare the device for the accurate dosing. We have to check the dose counter and shake the inhaler if needed, or maybe sometimes a priming is also required if it is a handheld device. The priming is nothing but to make sure that the device works properly in a uniform way and offers a uniform spray pattern with the given required quantity. The third most important part is the exhaling of by the subject. So we have to train subject how to exhale gently away from the mouthpiece and make themselves ready for the administration. Now, the technique of device in the mouth for administration is equally important. They have to keep the mouthpiece in the mouth till the chin and close the lips gently and tightly around the mouthpiece. Make sure there is no leakage or they do not inhale any additional air. After the mouthpiece is properly set up in the cavity, the inhalation technique by a subject is also an important part, because it has to be very quick and deep for the DPI, and for PMDI it has to be slow and steady. So depends on the type of the device. We have to train the subjects that how they have to inhale the product or the drug. Now, after we inhale, let the patient or subject inhale the content, they have to hold the breath for few seconds, which is up to 5 seconds. And then after 5 seconds, they have to remove the inhaler from the mouth, and they have to hold the breath for up to 5 seconds, which is important for the contact of the content inside the cavity. And then how to close the inhaler, cap it properly, and repeat if necessary for multiple administration. So this whole technique can be mastered by proper training, by a lot of mock rounds before the actual conduct of the study, to make sure we get the same kind of drug administration in same quantity each time throughout the trial for better outcomes. These are some operational risk and proactive mitigation plan that we normally follow at Veeda. Can be true for all the sites. In terms of the patient eligibility for spirometry, which is the most important part of the study, because here patient normally, if it's a long duration trial, they will take the administrations at home. So, how to make patient eligible for the trial is important, because the variability uh, in the spirometry is one of the risk, because the results and interpretation within subject, within site, or within group at the site can vary substantially. We have to reduce that variation. After the reading is taken, the interpretation of the reading itself of the spirometry by the same reviewer is also important. And if the quality is poor and the best test report review is not matching with all the subsequent or previous readings, we have to disregard the reading, and that may lead to many ineligible patients or ineligible records. Sometimes patients or technicians are not very cooperative in terms of providing us uniform readings and uniform technique. Sometimes we have to use a correction factor also to come to the final result. Sometimes people use a wrong correction factor. Now, these are all risks at site or at home if the study is long duration and patients are taking the doses at home. There are certain mitigation plan that we can prepare in advance to make sure the quality is up to the mark. Especially the training of technician and site staff and patients, we can do it well in advance. We already discussed this. The training and qualification of the PFT technician is also equally important. And as long as possible, we have to have a central reader who interprets and records the data in a form. If we can make the flow diagram available at the site and give an access to patients of that flow diagram of each steps involved in the FEV1 assessment, that will help all the subjects and technician to follow uniform practice. And the spirometry should be performed as per the ATS or ERS standards. And we will only include the patients which are able to provide the readings based on the American Thoracic or European Respiratory Society requirements. So these are some of the mitigation plans that we can do before the study starts or even during the conduct of the study. Veeda have been performing a lot of such study in past. We have some active studies at the moment as well. Especially we have a regional experience at different site. These are some key silent features that we have as a team for operations at the site level. Most of the topics I have already covered. We have been practicing this for a long time now. So we normally have a very good success rates in terms of the study outcomes about the uniformity of the results. I think that was my last slide. Bhavika, if you want to wrap up with a quick note. Otherwise, for me, that was all in terms of the today's topic. Thank you.
Thank you so much, Dr. Hire. Now, moving on, we'll be proceeding ahead with a discussion where we are going to answer some really interesting questions posed by our audience. The first question is, what are the general challenges in comparative clinical endpoint study in dry powder inhalation drug? Dr. Hiren, can you please answer that?
Sorry if I was mute. Okay. Can you come again with the question, Bhavika?
Yes. So, my first question was, what are the general challenges in comparative clinical endpoint study in dry powder inhalation drug?
Okay, okay. I think some of them we already discussed, but let us go into more details in order to answer these questions to the audience. Um, the challenges are very extensive because of larger populations needed due to high screen fail ratio. We need to have multiple sites, and these kind of studies are normally more lengthier than the routine BAB studies. Collection of On top of it, collection of spirometry data is also a challenge because there may be different in equipment itself, the validity or the variability of the technique by technicians, by patients themselves at home, by the reader, and by the interpreter. Um, Sometimes the results are also affected by concomitant and rescue medications at site because these all are patient population in most of the clinical endpoint studies. The subject retention, Bhavika, is also another issue because these patients are very vulnerable and they do not want to, just abide by the protocol rules on top of their existing medical condition. So retention is another challenge, and an accurate record of all the symptoms and medications because they are taking a lot of other medications for their existing disease. So a site level cooperation is also equally a challenge. So these are all the challenges, Bhavika, and the mitigation or the remedy could be the evaluation of data and results is have to have a very uniform reader and interpreter at the central level. The study design itself should be robust enough to provide us the reproducible results. All the confounding variables that I discussed before will try to minimize by a proper training at site to the patient, to the technician, and using a similar device across all the sites. This can address many of the challenges and confounding variables. Um, and this can result in high quality and less biased data. While designing the study, we normally recommend to follow the product specific guidance if it is available for the design and dose and the endpoints. If not available, we usually follow the general guidance available at various regulatory agencies in terms of the design and endpoints. In the protocol, we have to make sure that we provide extensive details so that the study staff at site level do not have to reach out to us frequently for any clarifications. They directly can use the protocol to train their technician and patients. And we can also use some other forms of data collection, which is subject diary or technician diary. I think that's how we can minimize the problems we may face and the challenges we have for this kind of studies using the mitigation strategies. Yeah, Bhavika, I think that I hope that answers the question.
Yes, Dr. Hiren, that answers the question. My second question would be, what are the specific challenges of having FE1, FEV1, sorry for that, for spirometry assessment as an endpoint in asthma and COPD trials and mitigation strategies?
Mhm. Okay, so specifically talking about the FEV1 spirometry assessment and other endpoints which are related to FEV1, there are a lot of challenges basically at patient level as well as the device level. We'll start one by one. The list is very long, in my opinion, but at least cover the top four or five, which I can think of now. One is the submaximal inhalation or exhalation, and that can underreport the true spirometry results. For that, we have to train subject properly, make sure they are aware about the techniques to provide an accurate and reproducible spirometry results. They have to make sure they fill the lungs fully and taking the deepest possible breath at the beginning of the dose and while providing the FEV1 measurement. Now, even while providing the assessment of FEV1, there can be slight hesitation in blowing the air out of the lungs after the before the initial blast, because they are not used to it. It's not their routine practice. Because we are asking them to do it, they follow the protocol. Otherwise, routinely they don't do it. So we have to make them understand why it is required to provide an accurate and uniform um, blow blow air out of the blast. So we have to train the subjects for faster and immediate blast during the FEV1. The poor initial blast can also affect the spirometry measurement throughout the study. Now, some subjects, because they are not used to provide this kind of assessment, may cough during the first attempt or the subsequent attempts. That can actually, if they cough it after the first or second, they can also affect the overall reading of the FEV1. Also, some subjects are hesitant to completely exhale the breath during the results, during the assessment, that can falsely report the spirometry results. So these are all the variabilities, and sometimes subjects takes an extra breath um, during the assessment, either through their nose or around the sides of the mouthpiece. That's why it is important to cover the mouthpiece completely and tightly within your lips. The device actually has to be on the tongue. Sometimes space and keep it under the tongue by mistake, but actually it should be over the tongue to make sure it has the access to all the site. There are two other errors that can happen, which is a positive zero flow and negative zero flow error, which is a device limitation in a flow type spirometry. These errors can be omitted by a proper validation and qualification of the device and training of the subject. These are all the problems that the patient and the site can face. We can train the subject to keep blowing until we ask them to stop for the accurate FEV1 measurement. We have to ask them that the blow should be hard and fast, and they have to keep blowing out until we ask them to stop. To avoid the extra breath they may take during the assessment, we can use a nose clip and may instruct the subjects to keep the tight lip seal around the mouthpiece. And for the device related errors, we can go for an accurate validation and a technique to make sure the errors are omitted. So, these are some of the things that I could recall now. There may be more in the list. However, these are the topmost that I can think of now based on experience I have. I hope that answers the question, Bhavika. Over to you.
Thank you so much. That was a very detailed answer, Dr. Ere. My next question is for Mr. Paresh. What are the major differences in FDA and EMA guidance for generic approval of OIDP?
Hi, Bhavika. So thank you for this question. And for this question, generally, we already presented in our slide deck that FDA is follow weight-based approach where we have to prove bioequivalence in terms of formulation, device similarity, in vitro test equivalence, as well as PK equivalence study and PD or clinical endpoint study. If we meet all these four criteria, then and only we can get the product bioequivalence. While in European submission, it is a stepwise approach where we have to prove first in vitro and device similarity. If we meet the first step itself, then we got the product approval. If we are not meet any one of the criteria for in vitro or device similarity, then we have to prove second step PK endpoint study. And in that PK endpoint study, we have to prove with and without charcoal block. And if we meet the step 1 and step 2, then we got the product approval. If any of the deviation in PK endpoint study or in vitro test, then we have to go for the step 3, where we have to perform clinical or PD endpoint study. So in European, generally less number of studies required as compared to the US FDA. So mostly currently people are prefer European submission as a first priority. Once they got the product approval in Europe, then they can try for the US FDA submission, where large number of studies and more number of studies are required, and which is very lengthy and typical to meet the requirements. Thank you.
Thank you, sir. That was a very detailed answer. I would like to thank all my panelists for nicely answering most of our audience's questions. I'd like to take a pause over here. We understand this session has kept us all very well engaged throughout, but due to time constraints, we won't be able to take any further questions at this point. But please be rest assured that we'll be sending answers to your questions via emails. Also, we'll be sending across this recorded link of the webinar to your registered email IDs. Lastly, on behalf of Veeda Group, I would like to sincerely thank all of you here for taking out the time and being a part of this webinar. I'd also like to thank and express my gratitude to Dr. Hiren and Mr. Paresh Patel for being such a wonderful presenters and giving us all great lessons today. We at Vida Group earnestly look forward to host you all again. Till then, take care and stay safe, everyone. Thank you.
Thank you, Bhavika.
Thank you.